Welcome. Let's dive into our findings on lactate dehydrogenase role in metabolism, specifically its interaction with ATP. Lactate has three big roles in our body: promoting Krebs cycle, the utilization of lactate to produce glucose; Warburg effect, a pyruvate to lactate conversion catalyzed by LDH to regenerate NADH; and being a major fuel for TCA cycle after converted into pyruvate. Lactate dehydrogenase serves pivotal role in pyruvate metabolism by reversible conversion of lactate to pyruvate with the concurrent reduction of NAD+ to NADH, which enables vital cellular lactate utilization, especially by providing NADH for gluconeogenesis. In other words, LDH is the only way that lactate can be utilized in the cell. There are two isoforms of LDH, LDHA and LDHB, which shares 75 identical amino acid sequence and have conserved structures. However, they differ in their tissue distribution and their affinities for pyruvate. The essential difference between LDHA and LDHB is their kinetic differences. The diagram shows that LDHA has low and LDHB has high binding affinity for lactate, which means LDHA requires higher concentration of lactate to efficiently bind to the substrate, and opposite for LDHB. Also, LDHA has high and LDHB has low capacity for lactate, which means LDHA can process large amount of lactate when it binds, and opposite for LDHB, indicating LDHA is more efficient catalyzing the conversion of lactate to pyruvate. The isoforms have different tissue expression patterns, with LDHA being highly expressed in liver, and LDHB being the predominant isoform in the heart, implying different physiological roles. Recently, Dr. Hicks and Rotter developed mass spectrometry integrated with equilibrium dialysis for the discovery of allosteric systematically, called MIDAS. There are two chambers. When it's combined, protein cannot move the membrane. However, metabolites can. After letting it to reach in equilibrium concentration, metabolites binds to the protein, and protein gets denatured. After the denaturation, they ran mass spectrometry to measure the abundance of metabolites and compare the top and bottom chamber in order to check with metabolite is binding. Through MIDAS, they identified ATP acts as an allosteric inhibitor for LDHA, but not LDHB. Knowing that, they ran activity assays for LDHA and LDHB. Graph A shows the activity assay that the activity of LDHA is lower than that of LDHB, which indicates LDHB is insensitive on ATP, but LDHA is sensitive. Graph B shows DSF analysis, which illustrates the thermal stability of LDHA, and LDHB is opposite, indicating binding with ATP provides LDHA better tolerance on heat. DSF analysis allowed us to measure approximate binding affinity by giving us an apparent KD value. They showed that both LDHA and LDHB have the same apparent KD for ATP. Here, this is my hypothesis that during fasting, liver LDHA is inhibited by ATP, leading to reduced lactate production and increased glucose production via pyruvate-fueled gluconeogenesis. I'm going to start with the initial steps for my final hypothesis. In order to confirm the results, we ran activity assays for LDHA and LDHB showing same inhibition with Dr. Hicks's result. However, we created the chimera of LDHB, which is sensitive to ATP but have the same kinetic properties with LDHA, called LDHB A N-terminus and A middle. Surprisingly, LDHB and TAME showed sensitivity on ATP, indicating it has complete sensitivity to ATP while maintaining LDHB's kinetics. In the future studies, I would like to study what happens to pyruvate when LDHA is inhibited by ATP using mass spectrometry. I will analyze the mass spectrometry data to understand how the cell uses pyruvate when LDHA is inhibited. For example, is pyruvate used in citric acid cycle or to make glucose via gluconeogenesis? Thank you to the members of the Roder Lab, especially Aaron Leifer for mentoring and designing the experiment, and Dr. Roder and Hicks for their excellent mentorship. This work was supported by the University of Utah Office of Undergraduate Research through Undergraduate Research Opportunity Program. Thank you for your time.